Hi there! The purpose of this video is to show you how to extract DNA from stool for the detection of soil-transmitted helminths via qPCR. This is a workflow that has worked best for our group for some years now, but feel free to adjust as needed and use alternative kits that work best for the aim of your study. Under UK regulations, all soil-transmitted helminths found in stool are classified as human pathogen hazard group 2 under the Health and Safety Executive. Please check with your country and institute or university-specific regulations in regard to processing potential infectious stool and/or the handling and disposal of corrosive hazardous chemicals. Group 2 can cause human disease and may be a hazard to employees. It is unlikely to spread to the community, and there is usually effective prophylaxis or treatment available. Hazard Group 2 biological agents should be handled at containment level 2. This is a suggested but non-exhaustive list of equipment and reagents you will need for the DNA extractions from stool. In this part, I'm demonstrating how to extract DNA from stool using the Fast DNA Spin Kit for Soil from MP Biomedicals. I am demonstrating the extraction of 4 samples, but you can reach max capacity, meaning 24 samples in one go. Before you start, you need to remove the stool samples from the freezer for them to thaw on ice for about 30 minutes. Do not forget to also take out the extraction control. First, write the numbers of the extractions on the tubes. I would write them on the side and on top of the cap as a backup. If you do not have barcoded stool samples, it would also be helpful to write the same extraction number on the original stool pot so it can be easily traced back if needed. In the lysing matrix tubes that contain glass, silica, and ceramic beads, you add 978 microliters of PBS and 122 microliters of MT lysis buffer. Make sure you always write the date when you open each reagent. Feel free to rearrange the workspace so it can fit your needs. It's best not to hover over the open tubes. If you can fit the clean ice bucket under the biosafety cabinet, then do so. If not, move each tube from outside separately. If the stool in a sample is relatively liquidy and does not have any preservative, then add approximately 70 to 100 microliters of stool, depending on the consistency. Cutting the bottom of the P200 tip might be helpful with aspiration. As mentioned earlier, if the original stool tube has ethanol in it and the tube can be centrifuged, it's best to spin it down, suggesting 10,000 G for 10 minutes, wash it with 2 times the volume of PBS, and then remove approximately 50 milligrams out of the stool pod using a sterile loop. Visit the SOP for more details. A scoopful of stool using the sterile inoculation loop is approximately 50 milligrams of feces. Use this approximation for all sampling. If the stool is sticky, you can twirl the sterile loop inside the tube and mix it with beads for quicker release. Repeat for all samples. Once you are done with loading all the lysing tubes with stool, ensure the cup top is screwed tight and transport the tubes to the homogenizer. The protocol we use is 40 seconds using the FastPrep 5G instrument on a speed setting of 6.0. You can use the saved Alpova wheat tissue setting as well for convenience. Once the homogenizer is done, remove the lysing tubes and place them in the centrifuge for a spin at 14,000 G for 10 minutes. If at all possible, it is good practice to change gloves after handling the stool to prevent any future contamination issues. While the centrifuge is running for 10 minutes, it's best to take advantage of the time to prepare the tubes for the next steps. During this time, you can prep the tubes for the protein precipitation and the binding matrix step. I'm only handling 4 stool extractions in this video, so if you're handling more samples, you might want to focus only on the protein precipitation step. For the PPS or protein precipitation step, take the same number of clear tubes out as the number of your samples and add 250 microliters of PPS solution in each new clear tube. If you still have enough time, you can prepare the tubes for the binding matrix step, but if there is not enough space, then leave this for later. It is best not to clutter the biosafety cabinet because this might affect the airflow. If you decide to proceed, like I'm doing here, for every sample, take 2 extra clear tubes and label them as your sample number A and B. Take the binding matrix from the box and make sure you vortex it vigorously as it precipitates rapidly. Be careful how you handle this. The matrix is extremely corrosive. After every sufficient vortex run, you can add 500 microliters of binding matrix from the bottom of the bottle in approximately 3 to 5 tubes. After you do 3 to 5 tubes, put the lid back on, vortex again, and repeat for all the samples. Put the prepared tubes with the binding matrix aside and proceed with the protein precipitation step, meaning those tubes containing the 250 microliters of PPS. Once the centrifuge is done, take the samples out of the centrifuge, and for each sample pipette the supernatant into the corresponding tube with the PPS. Once you are done, secure all samples to a microcentrifuge rack, seal them by parafilming over the entire rack, and invert 10 times by hand to mix. After you finish, place the tubes in the centrifuge for a spin at 14,000 G for 10 minutes. Take the extraction control out of the ice bucket and put it in one of the racks. In this step, you will also need your tube openers. Feel free to rearrange your space so you can feel more comfortable. Use a tube opener to open all tubes and add 1 microliter of the extraction control using a different tip per sample. Please be careful not to shake the supernatant after the addition of the control, as you do not want to move the pellet which contains all the proteins. After you finish adding the control, for each one of those samples containing the protein pellet, you will split their supernatant into the 2 tubes per sample you prepared and that are already loaded with a binding matrix. Note that the supernatant volume per tube may vary, but do not worry. See protocol for clarity and more details on the sequence of events. After you finish transferring all the supernatants to their respective binding matrix tubes, close the lids, apply parafilm to secure, and slowly hand rock the tubes for 2 minutes. After the time is up, remove parafilm and let the tubes rest for 5 minutes at room temperature. The rocking step allows for all the DNA swimming in the supernatant to be captured by the matrix. The resting step allows for the matrix to rest at the bottom of the tube so you can easily pipette out and discard most of the liquid at the next step. In this next step, you will use the spin filters to hold the binding matrix above the filter and remove the rest of the liquid at the collection tubes. For this purpose, take the spin filter columns out of the kit and label them with the sample numbers. Also, don't forget to change gloves frequently when possible. After the 5 minutes are up, use tube openers to carefully open all the tubes. At this stage, you remove the supernatant almost entirely, but leave behind approximately 1 millimeter of liquid, so you can aspirate the matrix and put it in the spin filter for every sample. Your matrix at this stage contains all the DNA found in the stool sample. At this step, the matrix from both tubes A and B for the same sample should be combined into a single spin filter with a collection tube. Use different tips between samples, but the same tip for A and B of the same sample number. Do not forget to visit the protocol for the full narrative and explanation of the steps. Lastly, don't forget to follow university or institute regulations regarding the disposal of the binding matrix. Once you're finished, close the lids of the tubes and place them in the centrifuge for a spin at 14,000 G for 2 minutes. After the filter tubes are spun, prepare some paper towels in your workspace so you can dry out the tubes after discarding the liquid in the collection tube. Dispose the liquid according to your country's and university's regulations and gently tap each tube on clean paper towels to remove any residual liquid. The liquid volume per tube may vary slightly. Make sure you tap each tube in a new spot on the paper towel every time. After you are done with this step, you are going to wash the matrix with ethanol-based buffer called SUIS-M from any impurities carried over from stool, such as inhibitors. Please note, as stated in your protocol, the Suez M washing solution comes concentrated at 12 ml in the box, and you need to add 100 ml of molecular grade ethanol to it. Invert it and mix a few times before you use it. Every kit comes with its own concentrated buffer, but you can use the same one you prepare with fresh ethanol for a few extraction rounds. Add 500 microliters of the prepared Suez M in every sample, gently pipette up and down, and be careful not to pierce the membrane in the filter. I use the same tip to add the ethanol buffer to all the tubes from a distance without touching the filter, and then I use a different tip per sample to mix the matrix with the buffer. If you're anxious, use a different tip to add the buffer at every sample. After you are done, close the lids and take the samples for another spin. After you take the spin filters out, use the same tube opener to open the tubes and discard the liquid at the collection tube. Note that ethanol is flammable, so dispose accordingly. Pat down the tubes to a new set of paper towels if needed. Then close the lids and place the tubes to the centrifuge for another spin at 14,000 G for 2 minutes. This step helps to dry the matrix further, as residual ethanol can interfere with downstream molecular work like PCR. Once the spin is done, remove the tubes from the centrifuge and open them under the cabinet for the matrix to air dry for 5 minutes. Whilst the matrix is drying, you can prepare the final collection tubes for the elution of the DNA from the matrix. After 5 minutes, add 100 microliters of DES into each tube and mix the matrix whilst gently stirring with your pipette tip. Do not pipette up and down and do not poke the filter. Stir for a few seconds per sample. Use a separate tip for different samples. Close the lid and take the samples for the final spin. This is just a suggestion, but I always like cleaning between steps and tidying up my workspace. Seeing liquid at the bottom is always a good sign. Use the tube opener to open all the tubes. Discard the column in every tube. Close the lids and you're done. Happy extractions!